In this third part, I will use the finding of spike time-independent plasticity and the concept derived from spike time-independent plasticity to explain or to explore a possible perceptual learning and memory. Let me begin with this part by playing a game with you. Now, this game is to name that tune. I'm going to play two pieces of piano music, and I want you to figure out what pieces they are and who's the composer. So, tune 1. So, any guess? It's a classical music by a famous classical composer. Very few people would be able to guess it, actually. Let's hear the second tune. Now, I'm sure most of you are familiar with this tune now. This is by Beethoven for Elise. Now, when my colleague Li Zhang made this two pieces of music by computer, I was amazed about this difference. It turns out that these two tunes are the same tune on the same score, but the first tune was playing backwards, with the same interval, same opposite sequence. So that leads to the idea that the memory that is a temporal specific. When we learn this tune, when we were in our childhood, our memory for this tune is temporal specific. We remember the sequence. And like many other temporal, memory in life, temporal sequence of events is very important. Now, we also remember the interval between the notes, right? So that's the time interval of events. So we have two aspects of this sequence of events. The temporal sequence from beginning to end, that we remember for tune 2. There's a time interval, which is the same in both two. So now, in this part, I want to illustrate with example to investigate how an animal or the nervous system can learn the sequence of events and remembers it. Now, the first experiment is again using the visual system rather than the auditory system where this game is about. It's an equivalent system of Xenopus tadpole again. We project a moving bar image across the retina of the tectum of the Xenopus tadpole by projecting this bar through LCD screen to the eye of the tadpole on the microscope stage. So this sequence excitation on the retina from one side to the next, from left to right, a constant sequence excitation is equivalent to having a sequence of excitation of notes in your brain, in the auditory system. But this is in the visual system. Let's see how this, the visual system could learn to memorize a sequence of excitation by a moving bar. So here is a moving bar across the retina. And when we put an electrode in the tactile cell, in the brain, we detect this. As the bar is moving across, the cell fires action potentials. Now we're recording the synaptic currents now by voltage clamp recording. This actually represents the spiking of the postsynaptic cell, the cell we are recording, created by a moving bar across the screen from bottom up. Now, from left to right, the same cell now gives another barrage of activity. It's slightly different. And down, top down, another set of activity, right to left. The activity looks different because you're exciting different retinal ganglion cells in a different sequence, right? Because this is a sequence of events that are specific to the stimulus. But because the input from ganglion cells is different, so you get a different firing in the tactile cell you're recording. But the overall excitation of this cell are equivalent. So we estimate how much excitation this tactile cell is receiving by integrating the charge, in the flow of membrane currents. The integrated charge looks more or less the same. When we have 4 different bars, 4 different type of stimulus moving across, they create more or less equal excitation of the tactile cell. So there's no direction selectivity in the individual tactile neurons as determined by moving bar across in different direction. They are not sensitive to direction. They are all responsive. The cell is receiving from different ganglion cells receiving input, more or less, total input is more or less equal. So now the experiment is very simple. You might have already guessed. Is that if we now train the tadpole by repeatedly giving a moving bar in one direction, like playing music in one direction constantly for a long time, what would happen to the response of the cell? Here is what we get. Before exposing the tadpole to unidirectional moving bar, the response is more or less equal, the green bar. So now we train with a moving bar in one direction for repetitively for 40 times, and then test their response to 4 different directions. We found that the trained direction now gives you much larger response, actually more firing, more spikes firing. When you test with the direction that was trained. So the same cell, become direction selective after they were exposed to a particular direction of stimulus for 40 times. So here's the time course. The time course, the total response before training is at this level. After training, a period of a few minutes of moving bars stimulation, the response goes up and it continues to be high for almost 1 hour. So there's a long-term selective response to that trained stimulus. The cell remembers that direction, the trained direction, is the one that it should fire more spikes. Right? This is long-term memory for sequence. Now, the interesting thing for this experiment is that the cell, when you test this response, this increased response only occurs if you are testing with the same speed of the training. Right? So you move in the training speed. If you test with a fast speed, it doesn't show up. It has to test with the same speed of the training bar to give you the higher response. So it's a highly specific network input to the cell you're recording that now they become tuned to direction. Right? This is learning, right? Learning to remember a sequence and directional sequence. Now, next I want to show you another example here in the primary visual cortex of the rat. Now, before that experiment, I want to give you a Gedanken experiment, that is a thought experiment, how we might expect this to happen. Now, if we excite a group of cortical neurons in a sequence from left to right, as shown here, the red activity are firing from left to right. So the synapse that made from by the left cell to the right cell are firing in a sequence that the presynaptic cell are fired before postsynaptic cell because the activity going in one direction. So all the synapse, the connection from left to right become potentiated according to spike timing dependent plasticity. And in the same, by the same token, the connection from the right cell to the left cells are weakened because of the reversed timing. So the result of a sequential excitation of a cortical neuron by a, say, light stimulation will result in strengthened horizontal connections between cortical neurons in a particular direction. So after the training, now one would expect, if now one excites a cell, for example, the first cell in this sequence, it will be much easier for the cell to fire a sequence of firing along this same sequence of training. It will trigger a sequential spiking if we initiate it from the left side. In the meantime, the excitation from the right side, from the endpoint here, will not likely to trigger a sequential firing because the weakening of the synapse from the right to left. So this is a Gedanken experiment. We thought that this might be happening. Let's see whether we can actually demonstrate this in the real cortex. So to do this, we perform this in rat visual cortex, primary visual cortex, because we have technology now capable of recording a large group of cells with a microelectrode array, which are shown here, that can be implanted in the rat cortex. For example, here are 2 arrays of electrodes recording 16 cells in the rat brain in the primary visual cortex, V1. So these cells are recording, the cells recording have receptive field that can be mapped from the visual field. Right? One can map the receptive field as shown here. The receptor within the visual field, for the first cell here, the receptive field that's that active receptive field that causes excitation of this cell is located at this region. And you can see that the receptive field are located in an adjacent, in orderly fashion along the electrode array. So these two array of electrodes, 16 electrodes, are showing up their receptive fields in a topographically aligned array along the visual field. This is what you expect because we are recording nearby cells. So now we have a sequence of cells that can be excited by visual stimulus. So now if we move a visual stimulus in the visual field from top to down in one direction, the cells that are recorded, the 16 cells are recorded would fire in a sequence. Now this is the firing rate with time. So they'll fire this cell fire here early, and then gradually the cell gets fired later and later because as a moving stimulus going across this visual field, they are exciting different cells at different time, right? So you have a sequential firing. So here is illustrated here. So now we have a group of cells. These are the cells that are being fired. The cell that's being recorded that has a receptive field at different locations in the visual field. So now if we have a moving spot right now, starting from this point to, you have to move across the screen to, during training, conditioning. So you have a moving bar spot from here moving across. You see these flashes of the activity. The flashes, the white flashes, is proportional to the spiking of the cell. We converted that spiking into flashes, right? So firing a sweeping spot going from the starting point to end point triggers sequential spiking of cells that have receptive field along this path. This is the conditioning with moving spot, right? Remember our Dunking experiment. We are moving the spot across to excite a series of cells. So the connection made by the cells that have receptive field here to the other cells down the stream will be strengthened, while the reverse connection will be weakened, according to our spike time independent plasticity. So now, if this is the case, what would happen if now we test again, after conditioning with real moving spot, we now test with a stationary spot, only trigger at a starting point. So the next spot, this is still the conditioning, right? So we have a conditioning that move across the field. So we are strengthening the connections among this group of cells. Then now we are doing now the testing with stationary spot. So remember, see, we have only spot flash. No, the spot is not moving, but there's waves created. We have a spot, sometimes spot here, sometimes spot here. Stationary spot. It triggers activity in the cortex. Now the question is, this test spot, can the test spot now recall the sequence of firing along this path that was conditioned during the moving spot? Whether the firing of this group of cells are more in a sequence resembling the moving by induced sequence, while the endpoint trigger, which will, if they trigger a sequential spiking, they should not resemble they should have a reduced or does not reflect the sequence that was conditioned, because the connections in these directions are weakened. So here's the example with conditioning, post-conditioning, after conditioning, a recall of the sequential spiking triggered by the test stimulus at the start point, right? So we use a partial stimulus to see if we can trigger the sequence. Now, how do you see the difference? Well, before the conditioning, you can use the test spot to test. You see this is the 7 consecutive test. Every time we have a spot on a test spot, the set of cells that are firing along the trajectory, they are firing, triggered by the firing, but there's no real sequence. There seems to be random before the testing, before the conditioning. So the test spot did not trigger really a sequential firing of the cells. However, after conditioning with a moving spot, now you test with test spot. You can see now the test spot is applied, then you see the sequential spiking. The spiking starts with a wave, right? So the closer to the starting spot, the cell will fire, and then the next cell, next cell. Now the second trial, it didn't show up clearly. The third trial, yes, again, it's sort of sequential. Fourth didn't show up. Fifth, sixth, seventh, all this trial shows there are more sequential firing resembling the training stimulus. Now one can do a much more elaborate analysis of the sequential firing by doing the correlation analysis to see this, how the so-called Spearman correlation coefficient, how the sequence of firing correlates with the sequence of positioning of the receptive field. And that correlation show a clear increase, a statistically increased correlation coefficient after training, conditioning. Now one can do another analysis about correlation between the firing of all the pairs of cells before and after their conditioning, and look at the difference. And difference show that there's a clear increased correlation that has a distribution of correlation proportional to the distance between receptive field. That is, there's a slant of this correlation that resembling the correlation with their distance, longer distance, their correlation is shifted more toward the right. So this slant of correlation represent, in fact, the increased sequential firing after conditioning as compared to before conditioning. Right? So this effect of increased recall of sequencing, a sequence of event, lasts for a while on the short memory, not too long. In fact, within 5 minutes, it decays to nearly equal level. And this we call a short-term persistence of a facilitated recall of sequence spiking. And this is consistent, entirely consistent with spike time independent plasticity in the intracortical connections within the primary visual cortex. So let's return to the idea of HAPS cell assembly that we talked about in the first part. Remember that HAPS says, how do we perceive perceptual memory? The perceptual experience is responsible for the formation of a cell assembly. Namely that the perceptual experience, say, such a stimulus, of a ring stimulus, it excites a group of cells in the brain, specific group of cells, and making their connection among them to be strengthened. So after the connection is strengthened, this self-assembly is established. Then one can recall this assembly by a partial stimulus that trigger only a small part of the assembly, and that can in turn, through the enhanced connectivity between them, among them, you have a recall of the entire cell assembly. So formation of perceptual memory and recall perceptual memory from a specific ensemble of cells through the connectivity, through the enhanced connectivity among them. This is the idea of HAPS cell assembly. And through the sequential learning experiment, we have exactly reproduced the idea, or tested the idea, that the sequential excitation of a group of neurons in the cortex will facilitate the recall of that same sequence by a partial stimulus, a start stimulus. Now wake up the stored memory, short-term memory of that sequence. So in this part, we have showed two examples, both sequence learning. The first example using Xenopus tadpole optic tectum, where I show that moving bar across the receptive field of the retina can rapidly train a specific cell to respond better to that moving bar. The cell can learn to respond to the sequence of firing of retinal ganglion cells in a specific direction. And in a set of experiments, I didn't go into detail, that this learning of direction selectivity depends on the spike time independent long-term potentiation and depression of the retinal tactile synapse. So that experiment will show the sensory experience induce refinement of perceptual property of a neural circuit. A property of circuit I can perceive direction of the visual stimulus. And that development of that property, perceptual property, depends on synaptic modification, the LTP and LTD. So in the second example, in a mammalian visual cortex, we showed this moving spot induced sequence firing, sequence spiking among the cells. That sequence spiking leaves its imprint, a short-term memory of that perceptual experience, and that experience can be recalled within 5 minutes by a partial stimulus of a spot stimulus at the start point of that spot stimuli.